we get started. So it's my pleasure to introduce Dr. Louise Moncla. She's an alumna of Fox Chapel High School, which shows her Pittsburgh roots. She did her undergrad degree at Penn State, got her PhD at University of Wisconsin in viral evolution. She has been a postdoc at the Hutch with Trevor Bedford in 2017. And she's a recent K99 ROO awardee on factors driving avian spillover. And she's going to be talking today about respiratory virus emergence and transmission. So Louise, the floor is yours.
Okay, thank you so much. And thank you everyone who's joining. I'm really excited to tell you about some of the work I've been doing in my postdoc, as well as some ongoing and future research I've planned, trying to understand the factors that drive respiratory virus emergence and transmission. Okay. Viruses are constantly emerging and reemerging. In April of this year, this article is in the science section of the
Oh no.
Oh. Are people still here?
That's weird. Yes, please, that'd be great.
How many people we might just not be able to see? Can you tell how many people are on the call?
Yeah, I can still see a couple people. If people are still on the call, could somebody say something?
I am here. This is Seema.
Yes.
Yeah, we're all still here.
Okay.
Okay, then
Great.
we keep going. All right.
All right, I'll keep going then. We thought we lost people. All right, so this article was in the New York Times this April. A new bird flu jumps to humans. COVID fuels the current virus pandemic, but the world is full of flu viruses waiting in the wings. So this article is describing the first reported human case of a new type of avian influenza that was found in humans for the first time this winter. This was H5N8. There were 7 people in a poultry plant in Russia who were infected. And although the outbreak didn't spread beyond the plant, 100,000 chickens died, and an additional 800,000 chickens were killed to prevent this outbreak from spreading. Now, this news was largely buried at the time because this was right around the time that the coronavirus variants were starting to emerge. But this outbreak itself was part of a larger series of outbreaks that were pretty incredible that were happening this winter in Europe and Asia. And I think that this is a really shocking statistic. During the period of time between December 25th, 2020 and January 14th of 2021, so about 3 weeks, 7 million birds died of H5N1 across Europe and Asia. The bird flu situation this year was actually so crazy that the Wall Street Journal wrote an entire article that was just called "What is Going On with Bird Flu?" So, even though we haven't really heard much about it, it's been a pretty crazy year for avian influenza. Beyond the emergence of these novel viruses, we've also seen the recent resurgence of previously well-controlled vaccine-preventable diseases. If you can dig deep into the recesses of your memories to the before times, you may recall that we had a series of pretty bad measles outbreaks in 2019. So these outbreaks occurred in New York, as well as in Washington State, where I currently live. Mumps has also made a comeback. Mumps has been causing recurrent outbreaks since 2006, which I'll talk about in detail later in my talk today. But more recent mumps outbreaks have caused these headlines from 2016 and 2018, in which entire sports teams have had to sit out of games. And I'll come back to this theme of kind of college outbreaks later in the talk as well. But while I was making the slides for this presentation, I came across this really excellent photo actually from Pittsburgh. So in 2014, there were a series of very widely publicized outbreaks in the National Hockey League. And I remember this happening. Very famously, Sidney Crosby of the Pittsburgh Penguins got the mumps, had to sit out of the game, and there were some very unhappy Pittsburghers. So when viruses emerge, and when new viruses emerge, when old viruses make a comeback, we want to know, why is this happening now? How can we stop transmission? And how can we prevent new outbreaks from occurring in the future? But of course, answering these questions is not really straightforward because viral emergence and transmission is complex. There are lots of different factors that determine how and why new viruses enter our population. Most of the new viruses we encounter are zoonotic, meaning that they come from animal reservoirs. Humans are constantly coming in contact with animals and by extension, their viruses. And the ability of these viruses to infect new host species like humans and transmit efficiently depends on a lot of different factors coming together. It depends on virologic factors like the ability to bind host receptors and interact with host machinery and intrinsic antiviral factors. But it also depends on both ecologic and epidemiologic factors like habitat encroachment, husbandry practices, and how humans and animals interact with each other. Once these viruses are established in human populations, transmission intensity then depends on a number of other factors like population immunity, vaccination rates, and human travel, interaction, and movement patterns. So understanding the factors that drive viral emergence and transmission is critical for designing public health interventions and surveillance practices. But doing so requires a tool that can capture how all of these different processes have shaped viral transmission in the past. And one tool that does that pretty well is genomics. And the reason is that viral genomes contain a record of a virus's transmission history. So I'm just going to walk through a quick example here where I explain to you exactly what I mean by this. So here I'm going to use the single green dot to represent a single person who's infected with an RNA virus. Okay, so let's pretend this person's infected with flu. This person's going to go through the course of their infection, and let's say that they interact and infect 2 more people. Let's say 1 at 1 day post-infection, 1 at 5 days post-infection. Because all of the viruses I'll be talking about today are RNA viruses, they all have high mutation rates and quick generation times. And what this means is that mutations accumulate during this transmission process as we move forward in time. So I'm going to be using this kind of exploding yellow star here to represent a mutation. And as we go from left to right across the screen, that'll be going forward in time. Okay, so if we were to follow this entire transmission process, and eventually if we sampled every single person who was ever infected in this outbreak, we could reconstruct a full genealogy of how that virus spread through that population. Of course, in reality, we never sample everyone. We don't even come close. But it turns out that even if we sample only a fraction of individuals, we can still use those genomes to reconstruct a tree that accurately shows the relationships among the sampled members of that phylogeny. And because viruses accumulate mutations at a constant rate over time, these genomes then contain a record of the transmission process, even when every single transmission event wasn't directly sampled. We can then start to layer other types of data on top of these trees to make inferences about transmission timing and directionality. So for example, if we had date information, we could make inferences like all of these infections shared a common ancestor back in October of 2020. And if we had other information like host sample, for example, if this weren't actually an avian flu tree, we could make inferences like this, where we would infer that there was an avian to human transmission event sometime in October of 2020. Finally, because of course we also have mutations on these trees, we can then start to look for mutations that co-occur with transmission events of interest. So for example, we might find that this mutation here actually elicited a switch to human type receptor binding. Now in this scenario, we might want to go one step further. We might wonder, where did this mutation arise? Did it arise in the bird or in the person, and what happened there? And for questions like this, we can use genomics to look at within host diversity. Because RNA viruses in particular have such high mutation rates and such quick generation times, when you're infected with one of these viruses, you're actually infected with an entire population of virions that harbor genetic diversity. We can characterize that genetic diversity by taking a sample of it and deep sequencing it. And when we do this, we not only get information about the consensus genome, which we can use to build a phylogeny, but we also get all this other information about the number and frequency of polymorphisms that exist within these infected people. So genomics across scales can be used to disentangle the evolutionary and epidemiologic factors that drive viral emergence and transmission. Today, I'm going to be focused on these 2 big overarching questions. How do viruses cross species barriers, and how do viruses move across time, space, and population groups? I'm going to tell you about how we've been trying to answer these questions using viruses that are both emerging and viruses that are endemic. I'll start off by telling you about the emergence of H5N1 avian influenza viruses. I'm going to tell you about some previously published work where we looked at within host evolution in humans and poultry during natural H5N1 spillover, and about some current and ongoing work where we're trying to estimate rates of H5N1 cross-species transmission. I'm then going to switch gears and tell you how we can use similar tools to answer really different questions about transmission of established human respiratory viruses. I'm going to tell you about a study that we published this spring where we looked at mumps transmission during an outbreak in Washington State in 2016, and how that's led to some future work on health disparities and sourcing dynamics. Okay, So I'm going to start off with the emergence of H5N1 avian influenza viruses. I'm going to tell you why we care about these viruses and what some of the open questions in the field are. Like all influenza viruses, H5N1 avian influenza viruses naturally circulate in wild aquatic birds. This is a photograph of migratory ducks flying over a lake in Myanmar. Since its emergence in 1996, H5N1 viruses have caused recurrent outbreaks in humans and birds. Influenza viruses' natural hosts are these wild migratory birds, and genetic diversity is shuffled when these birds stop at these kind of stopover sites like lakes and interact with each other. And because these natural hosts are migrating birds, these viruses can be spread over really long geographic distances, including across continents. And in fact, if we look at the global range of where avian flu has been found, specifically H5N1, we see that it's been found all over the place. So this is a screenshot of the H5N1 NextStrain build that I currently maintain. So for anyone who's unfamiliar, NextStrain is a website for real-time tracking of viral pathogens that shows phylogenetic trees along with maps. So the size of the dots here represent the number of genomes that have been sampled from a given country. So as you can see, H5N1 has been found all over the place, with particularly high rates of incidence in Asia. Although wild birds are the natural reservoir for flu, these viruses can also infect domestic birds, and H5N1 has actually become endemic in some domestic bird populations. And when these viruses integrate into poultry populations, they really increase the risk for human infection. Humans are generally thought to be infected by bird flu from interaction directly with sick poultry, and this is often facilitated by interaction at these live bird markets. When humans are infected with H5N1, case fatality rates can be as high as about 60%. Currently, humans are thought of as dead-end hosts for avian flu, meaning that these viruses can infect humans, but they don't generally transmit very well from person to person. However, there's some concern that if these infections continue to happen periodically, that one day these viruses could evolve to transmit efficiently among people. And concern for this possibility was heightened in 2012 when these two papers came out. So what Amai et al. And Hurst et al. Did was they took an avian virus and they passaged it in ferrets until it could replicate and transmit efficiently. Ferrets are the most common model for human flu transmission. And what they found was that only 3 or 4 amino acid changes were required to render these viruses highly transmissible. And these mutations tended to occur in the receptor binding protein and in the polymerase complex. So this was sort of a worrisome finding when it came out. But these studies left a big open question. And that was, does this actually happen in nature? So when humans are actually infected in nature with these viruses, do these viruses evolve in those people? Do these viruses acquire human adapting mutations within host? So in collaboration with the Pasteur Institute in Cambodia, we sought to answer this question. So H5N1 is endemic in Cambodia, and the Pasteur Institute has been doing surveillance there for a really long time. In 2013, they had an outbreak where they sampled humans and ducks who were naturally infected with H5N1, and these were domestic ducks. And they sequenced these viruses on an aluminum ice-seq. What was exciting about this data set was that although there had been a couple of studies that were small that kind of looked at within-host diversity in humans, nobody before this had ever looked at within-host diversity in naturally infected birds. So it was nice to be able to look at how evolution proceeded in humans compared to a more natural host species. Our hypothesis here was that human adapting mutations should arise within naturally infected humans. And the first thing we did to try to address this was that we just looked at every mutation that occurred within hosts across the genome. So what I'll be showing you here is on the x-axis, the nucleotide site across these 10 open reading frames that we looked at. And on the y-axis is the within host single nucleotide variant frequency. I'll be showing you human mutations in circles and duck mutations in squares. Okay, so when we looked across the genome, we found mutations across all of these 10 open reading frames in both humans and in ducks. We did a bunch of analyses to analyze this diversity and look for natural selection, and I don't have time to tell you about all those analyses today. But I do want to tell you about a couple of interesting things we found. So one thing we looked for was whether we ever found these mutations that have been previously identified to elicit human adaptive phenotypes. So I'm now going to highlight with colors here mutations that have been previously shown to elicit these adaptive changes. Okay, what I'm showing in red are mutations that arose in humans, and in blue, mutations that arose in ducks. And as you can see, we found that in humans, but not in ducks, we found mutations that have been previously identified as being adaptive. Of particular interest, we found a number of mutations in the hemagglutinin or receptor binding protein that have been shown to elicit a switch in receptor binding preference. So we found this alanine 150 valine mutation in 2 separate individuals. This has been previously linked to a switch from avian type to human type receptor binding preference and was also identified in a different study of infected individuals in Vietnam. We also found this glutamine 238 leucine mutation. And this mutation is interesting because it was one of the mutations that was identified in those original ferret transmission studies as being important for transmission. We also found in a third patient an arginine at this site. And although there's nothing known phenotypically about an arginine here, it's interesting that we observe so much diversity. We also identified this other mutation in the polymerase complex, particularly this PB2E627K mutation. This mutation has been identified repeatedly in both laboratory and natural infection studies, and it's known to really improve avian virus replication in humans. What's interesting about this mutation is that although we found it in one person, we only found it in one person. This is a mutation that has been hypothesized to be almost necessary for human replication. And in fact, a lot of the these ferret studies don't work very well until you add this mutation into the viral backbone. So it's interesting that we found this mutation at a low frequency and so rarely in general. So we found that H5N1 avian influenza viruses do generate putative human adapted mutations during natural spillover infections. But we also found that these mutations stayed at low frequencies over time, and that most infections that we looked at actually lacked a known host adapting mutation. The mutations I just highlighted for you, we would have predicted for them to have been very strongly selected within host after weeks of replication in the upper respiratory tract. But they were all present at low frequencies. Most of the infections we looked at in our study also didn't have a known adapted mutation either at consensus or within host. So when the study was finished, we published it and I went to go visit our collaborators in Cambodia. And I started to get really interested in this question of, which factors beyond these laboratory identified mutations are important for H5N1 cross-species transmission? So I spent a bunch of time interviewing people in both Cambodia and Vietnam who are doing avian flu surveillance. And I came to realize that although avian flu is a really well-studied pathogen, there's actually a lot about this process we don't understand very well. I think the virologic studies have done a really good job telling us about how vaguely avian viruses can, a couple pathways they can take to becoming vaguely human viruses. And we have a general sense that viruses, are transmitted from wild birds into domestic birds and then domestic birds into people. But it turns out that a lot of these details are actually not very well worked out. For example, although we know that viruses are transmitted from wild birds into domestic birds, nobody's actually estimated how frequently that happens or if there are geographic locations where that happens more often. We also don't know if there are particular environmental or husbandry practices that allow this to happen more efficiently. Similarly, once these viruses get into poultry, it's actually not very well understood why some of these lineages become endemic. Are they sustained by poultry trade? Is there a seasonal component? And some of these questions actually came up a couple weeks ago when I was talking to my collaborators in Cambodia. They were telling me that they'll often find new lineages of H5N1 in their poultry populations, and they have no idea whether that's the result of a new introduction from wild birds or whether it's from illegal poultry trading across neighboring countries. Finally, there's this big open question of why only some of these viruses that successfully infect poultry actually infect people. There have been 17 clades of H5N1 avian flu virus that has infected poultry, and only 5 have ever caused human infections. And what's interesting is that this does not always break down along which of these have these known adapted mutations versus those that do not. So I decided that this was an interesting area with lots of important open questions, and I wanted to try to answer some of these. So I wrote a grant to try to answer some of these questions, and I decided that the first thing I wanted to do was to try to get a handle on how frequently cross-species transmission occurs between wild birds, domestic birds, and humans. So I'd like to tell you a bit about some of the data we have now and why I think getting a good handle on this question is going to really set us up to be able to answer some interesting questions going forward. So the first step here was to generate a data set. And so I'd be happy to talk to you about exactly how I did this in more detail afterwards if you're interested. But basically, using a combination of publicly available data and doing a bunch of manual curation and kind of sanitizing this data set, I ended up with full genome sequences from about 400 wild birds, about 2,000 domestic birds, and about 300 humans, going from 1996 until present. I did some subsampling to even out the host and geographic representation, and then I applied what's called a structured coalescent model. And what these models do is they estimate the population size in each group and the transmission rate between them. So our hypothesis here was that there will be lots of introductions of H5N1 from wild birds into domestic birds, that some of those introductions will lead to long-term circulation in poultry, that humans will be primarily infected from domestic birds, but that we will not observe long-term circulation in humans. Okay, so when we ran this model, this is the phylogenetic tree that we got. So what I'm showing you here is a time-resolved phylogeny. On the x-axis is the time in years. So for each tip, that represents the date that the sample was collected, and for each internal node, that represents the inferred time to the most recent common ancestor. On this phylogeny, we've reconstructed these internal node states. So the color here represents the posterior probability that that internal node is within that given group. So as we walk down this tree, you'll notice that the entire backbone of the tree is inferred with pretty high probability of being within wild birds. And that tells us that H5N1 is primarily maintained in wild birds. And this makes sense. This is what we kind of know about H5N1 epidemiology. But as we walk down the tree, we see that there are instances in which the backbone color switches into blue. And those indicate events where we had a transmission event from wild birds into domestic birds. What you can see is that there's some introductions that lead to very long-term circulation. So there's these large lineages that are dotted with human infection shown in red. But there's also a bunch of introductions that don't lead to long-term circulation. So if we want to look at this a little bit more in detail, I'm going to show you now a different view of this tree that we're calling an exploded tree view. And what I've done here is I've separated out every introduction into domestic birds as its own little mini phylogeny. So when we look at the tree this way, what I'm showing you here is the color of the large dots to the left represent the source of the introduction. In this case, they're all from wild birds in yellow. And what we can see when we look at this view is that there have been lots of introductions from wild birds into domestic birds, and that these are happening at a reasonably constant rate over time. We see that most of these introductions lead to only one or a couple sample tips in domestic birds, but that a small subset lead to these much larger, long-lived outbreaks. We can actually count the size of these introductions in terms of the number of tips. So that's what I'm
Can I ask
showing
a
you
question
here.
on the previous?
Yeah, sure.
I'm just curious, you show closely related nodes there, but how closely related are they? The ones that are right next to each other, are they highly related or?
Do you mean close together on the y-axis?
On the y-axis, yeah.
Yeah, so the y-axis as here is actually ordered kind of by date. So, it's kind of just as you walk down the tree in this way. So it's a little bit difficult to say how close they are without looking at the full tree.
I see.
Because this doesn't capture information about the branch length leading up to those. Yeah. Okay. So if we look at the size of these introductions in terms of the number of tips, we see that the vast majority of introductions lead to kind of one or a couple sample tips, but that a subset lead to these much larger long-lived clusters. And finally, if we quantify the period of time that these introductions circulate following an introduction, we see that most introductions from wild birds lead to only short-term circulation of less than a year, but that a subset lead to much longer-term circulation, sometimes up to about 10 years. Okay, so some key takeaways from this part of the talk. I showed you that H5N1 viruses generate putative human adaptive mutations during natural spillover infections. I showed you that H5N1 cross-species transmission can be modeled with a phylogenetic tree. Preliminary data shows that H5N1 is primarily maintained by transmission within wild birds, with frequent spillovers into domestic birds. And that while most introductions into domestic birds lead to small, short-lived clusters, a subset do lead to long-term circulation. So in generating this preliminary data, I'm pretty excited to have all these models worked out and working really well, because this can now be really easily expanded to address a lot of interesting questions. An obvious follow-up here is to figure out what's going on with these long-term poultry lineages. So, an obvious follow-up is to look at which environmental and husbandry practices correlate with cross-species transmission and long-term circulation in poultry. The idea here is to pull in factors like poultry production volumes, human population density, and we can actually plug these right into the phylogenetic model and get an effect size for each of those different predictors. Another thing I'd like to do is look at which mutations are preceding successful human infections on the tree. So we actually have mutations mapped onto this same phylogeny. And so we can go back and look and see, which mutations are occurring on these host transition events, and then we can phenotypically validate those mutations to see whether they cause phenotypes that make sense. Today, I've only showed you information for one gene in the genome, the receptor binding protein. We'd like to expand this across the entire genome and potentially look at patterns of reassortment. And the ultimate goal of all of this is to see whether we can use this information to improve surveillance and vaccine designs. Okay, so far I've told you about how we can use genomic tools to understand, how novel zoonotic viruses emerge using H5N1 as a model system. I'd now like to switch gears and tell you about how we can use some of these same tools to answer really different questions about how endemic viruses are transmitted among people. I'm going to spend most of the time telling you about a project we did where we looked at mumps transmission during an outbreak in Washington state, and how that's led to some future work I have planned on health disparities and sourcing dynamics. All right, so prior to the introduction of the 2-dose MMR vaccine in 1989, mumps was a really common childhood illness. This is an actual photograph taken from 1957 of a kid named Greg Cox who got the mumps. Pre-vaccine, it was estimated that about over 90% of US children had experienced mumps by the age of 20, and the vast majority of cases every year were in children. So the introduction of the MMR vaccine in 1989 was this amazing thing for public health. And incidence of mumps in the US plummeted, and it remained really, really low for a long time. Until 2006. And all of a sudden in 2006, since then, we've seen these recurrent outbreaks of mumps across the United States. This culminated in 2016 and 2017 with the largest mumps outbreaks in the post-vaccine era that we've had. Now, the question I always get right off the bat is, can we blame this on vaccine hesitancy? And what's interesting about mumps as compared to measles is that most of these outbreaks have had two big things in common. The first is that the majority of these cases were in vaccinated individuals. And the second is that most of the cases were actually in young adults. So this phenomenon was explained really well by this paper published by Joe Leonard in 2018. Basically, what he showed is that immunity to the mumps vaccine wanes over the course of about 13 to 30 years. And that if you incorporate this waning immunity into these epidemic models, it really captures nicely the timing and age distributions of these outbreaks. So that's great. That explains, a lot about what's happening in these college-associated outbreaks of mumps. But it actually can't explain what happened in Washington. And that's because Washington's outbreak was different in two key ways. The first thing that made our outbreak a little weird was that Washington state experienced a really high incidence rates in this one particular close-knit ethnic community. So if right off the bat you're wondering who this community is and what is going on there, I promise I'm going to come back to this at the end of the talk and give you lots of context. But for now, our outbreak began in October of 2016 and ended in the spring of 2017. We had 891 total cases in our outbreak, and about 52% of our cases came from this community that comprises less than 1% of the total population in Washington. And before you ask, this community actually has higher overall vaccination rates than the general public. So we don't think that this is related to vaccination. The other thing that made our outbreak a little weird was that most of our cases were actually reported in kids. So our highest incidence rates were in 10 to 13-year-olds and 14 to 18-year-olds. What's even stranger is that the exact same thing happened in Arkansas. So Arkansas was unique because it was actually the largest outbreak of any of the outbreaks in 2016, 2017. 58% of Arkansas's cases came from the same close-knit ethnic community. And their highest rates of infections were also in 5 to 17-year-olds. So the Washington State Department of Health had done an outbreak investigation. But there are a few questions that were open at the end that we were still not sure about. So we wanted to know, how was mumps introduced into Washington and what drove transmission within the state? We wanted to know in terms of introductions, how many times mumps was introduced and from where. So we wanted to know, were there introductions from neighboring states like Oregon or British Columbia, or was there something more interesting going on? In terms of what drove transmission in the state, we were really interested in whether people who belong to different epidemiologic groups were transmitting differently. So we collaborated with the Washington State Department of Health, and we sequenced about 166 genomes from Washington and other US states. For each of our genomes from Washington, we knew their vaccination status, whether they belong to this ethnic community, the date that their sample was collected, and their age. So we combined these genomes with publicly available genomes from throughout all of North America, and we built a phylogenetic tree. So what I'm showing you here is another time resolved phylogeny. Each tip represents one unique sampled mumps genome. The x-axis represents time. That's the date of sample collection, and for the internal nodes, that's the time of most recent common ancestor. Every color here represents a different US state or Canadian province. And I've highlighted our Washington genomes with these blue dots with these arrows. Okay, so when we look at this tree, right off the bat, what you can see is that our Washington genomes fall in all across the diversity of these North American genomes. And that tells us that mumps was introduced into Washington multiple independent times. We can make that inference because these genomes at the top that are inferred from Washington share a common ancestor with viruses from another geographic location before they share a common ancestor with other viruses from Washington. If we want to know exactly where these introductions came from, it's a little bit easier to look at if I bring back that exploded tree view. So I've now again separated out each introduction of mumps into Washington and plotted it separately. Again, the large dots on the left represent the inferred location that these introductions were seeded from. So when we look at the tree this way, we see this cluster of highly divergent genomes at the top of this slide. And we actually have to trace those branches back all the way to 1999 before they share a common ancestor with these viruses. We think that these likely represent travel-related importations from geographic locations that we don't have very good sampling for. We then see this kind of smattering of introductions from Ontario and Missouri that lead to one or a couple sampled cases. And then we see that the majority of the tips in our tree come from these 4 introductions from Arkansas. So as a reminder, Arkansas has this very similar epidemiology to our outbreak. So it's interesting for us to identify a shared geographic link as well. You'll also notice that these introductions from Arkansas tend to see larger, long-lived transmission chains. So given that we could recover this relationship with Arkansas, we were next really interested in this question of what was driving transmission within the state. And what we wanted to know was whether infections in distinct epidemiologic groups were equally likely to be transmitted. And so we came up with this little transmission metric that uses a divergence tree. And the goal of this transmission metric is to try to assess whether groups of individuals are enriched at the beginnings of transmission chains. And the idea here is that individuals who are part of groups that are transmitting more on average should be present at the beginnings of these long, successful transmission chains, more so than groups of individuals who are not. The way this metric actually works is that we traverse the tree from root to tip, and when we encounter a tip that lies on an internal node, we then evaluate whether that tip has any descendants in the tree. And by descendants, I just mean kind of downstream branch mass as we move from left to right. So those top 2 tips do have descendants, so we labeled them as basal tips. And if they don't have descendants in the tree, we called them terminal tips. We've then evaluated the probability of being basal or having descendants as a combined function of community status, age, and vaccination status with a logistic regression model. We looked at vaccination status because we wanted to know whether individuals who had either an unknown vaccination status or who were unvaccinated were transmitting more on average. And we wanted to look at age because of this waning immunity hypothesis. This is the actual regression model. You'll notice that we have two vaccination statuses here because you could be either vaccinated or unvaccinated, and you could have either a known vaccination status or an unknown status. Okay, so when we ran this regression model, this is what we found. We found that community status, but not vaccination status or age, was a strong predictor for being upstream in these transmission chains. We estimate an odds ratio of about 3, meaning that community members were 3 times more likely to be found at the beginnings of these long, successful transmission chains than non-community members. And this suggests that community membership was a significant determinant of transmission while controlling for vaccination status and age. So we wanted to follow this up with a more robust metric for transmission. So we wanted to know kind of how much transmission was occurring within community versus non-community members, and how much transmission was occurring between them. There were sort of 2 hypotheses we wanted to assess here. The first is that, maybe there were actually just 2 separate outbreaks, and there wasn't much interaction between these groups. So that would be the hypothesis shown down. Our other hypothesis was that, maybe transmission is being sustained primarily in one group with kind of short terminal transmission chains when you have these introductions into the other group. These 2 hypotheses lead to really different hypothesized trees. So in hypothesis 1, where the outbreaks are separate, we would expect individuals from these 2 groups to kind of form their own clades on the phylogeny. On the hypothesis on the left, we would expect that most of the internal nodes on the phylogeny and most of the backbone should be within one group. And then when we observe transmission events into the other group, they should form these short terminal transmission chains. The reason we expect this is that internal nodes on a tree represent ancestors to subsequently sampled tips, while terminal nodes or tips represent infections for which we didn't sample any offspring. So to reconstruct these internal node states and estimate these transmission rates, we then brought back that structured coalescent model. So we took the 4 introductions into Washington that had both community and non-community member tips, and then we ran them through this model. And these are the trees that we get. So what I'm showing you here is the color of each tip represents whether it came from a community member in light blue or a non-community member in dark blue. The color of these backbones now represents the posterior probability of that internal node being within this community. So as you can see, we're reconstructing with pretty high probability that most of this transmission was occurring within the community, and then when we saw infections outside of the community, they led to very short terminal transmission chains. In this model, we get an estimate of the number of transmission events that occur in these different directions. As you see, we're inferring lots of transmission from community to non-community members, but very few in the opposite direction. And this tells us that transmission was predominantly occurring in one direction. So we did a bunch of other analyses to look at transmission and to control for sampling. What we kept coming back to was just that transmission was really efficient in this community. And, we were trying to figure out why this was, and we decided that the best way to answer this question was to talk to the community. So we sought out local community health advocates, and we connected with 3 individuals as well as a trusted community ally, and we shared the paper with them. We ended up then hiring on one woman as a consultant who was interested in consulting with us on the paper about how to contextualize our results and present our findings. So I want to spend the last little bit of this part of the talk giving you some information about who this community is, why they were so affected by mumps, and why I think the results we found here are likely applicable to other viruses as well. Okay, so this community is the Marshallese. The Marshall Islands are a series of low-lying islands in the South Pacific. The US forcibly occupied the Marshall Islands after World War II until 1986. And the Marshall Islands were viewed as this very strategic military base for the US. At the time, the US was also pretty interested in nuclear weapons testing. And between 1947 and 1986, the Marshall Islands served as the primary nuclear testing site for weapons in the United States. As part of this testing program, the US detonated the equivalent of over 7,000 Hiroshima-sized nuclear bombs, the largest of which took place during this Castle Bravo test, which took place on March 1st, 1954. As you can imagine, the effects of this testing were really devastating. There was widespread environmental destruction, and individuals were forcibly moved off of these islands, but even so, many were exposed to nuclear fallout that persists on these islands today. As part of reparations for this testing, the US signed this treaty in 1989 called the Compact of Free Association. And basically this stipulated that individuals could live and work in the United States without visas. However, healthcare access under Medicaid was revoked for Marshallese individuals in 1996, sort of due to a clerical error, actually. And this has been really bad because US-residing Marshallese tend to lack access to healthcare, particularly preventative medicine services. So the Marshallese are a really unique group in the US. They're this legal resident population who have been kind of somewhat forgotten by the US government. So if we weave all this information together, we can start to hear for why this group was so at risk for mumps. We've been thinking about mumps a lot in terms of this number called the force of infection, or the per capita rate at which susceptible individuals contract an infection. And the force of infection is a function of the number of people who are susceptible in your population, the number of infectious individuals, the total population size, and this beta parameter, which is the transmission rate. And the transmission rate is the product of the contact rate times the transmission probability. What we think is that in these college outbreaks, we're driving up the force of infection by creating more susceptible people from waning immunity. But you can also drive up infections without changing the number of people who are susceptible by altering the contact rate and transmission probability. So in thinking about why this community was so at risk, there are three big things we settled on from doing these interviews. The first is that there remains pretty significant concern within the Marshallese community regarding long-term immune damage from radiation exposure. Banked historical samples confirm uptake of radioactive materials in Marshallese inhabitants of affected islands. And although there's been really limited data kind of going into the mechanism for this, I think this really serves to underscore that Marshallese trust in US medical and research institutions was really decimated by this testing. And this is problematic because people feel that they are more susceptible to infectious diseases, and they're much more hesitant to work with public health. Another thing that we think is probably important here is contact networks. So multi-generational living is very common in the Marshallese community, and the average household size for a Marshallese family is larger than that of the overall American populace. So having more household contacts may have facilitated a greater number and higher intensity of interactions among individuals, allowing the force of infection to overcome pre-existing immunity. Our community advocate actually really agreed with this. She self-described the Marshallese community as really close-knit and agreed that probably housing size could play a role here. But the last factor that we think is probably important that hasn't really been discussed as much is healthcare access. Multiple studies have shown that Marshallese individuals will often forego medical care due to a lack of insurance. Other studies have shown that blame for poor Marshallese health outcomes is sometimes placed on host genetics, cultural practices, and poor health literacy, with much less consideration for how these attitudes and behaviors directly stem from US occupation. So hesitancy to seek healthcare could be important for increasing transmission if individuals are spending more time at home interacting with people and are reluctant to seek care. So some key takeaways from this part of the talk. We found that, in contrast to what we would have thought if we had just looked at case counts, our outbreak was comprised of multiple introductions from disparate locations, sparking overlapping transmission chains. The second big takeaway was that we need to really broaden our understanding of who is at risk for mumps. A lot of the conversation in mumps right now is really focused on identifying groups who are at risk due to their age and waning immunity. But what the outbreaks in Washington and Arkansas showed us was that other factors like high contact rates and poor healthcare access can allow an outbreak to be sustained in groups that are highly vaccinated and who we wouldn't expect to be at risk due to waning immunity. And our last big conclusion was that social networks can be the primary risk factor for a respiratory virus outbreak, even when a vaccine is effective and even when it's widely used. And we think that this is likely important for considering long-term SARS-CoV-2 practices, because it tells us that even a really good, effective vaccine, while really important, is not going to be a panacea for the disparities in disease risk that exist in our society. So there was a lot of interesting stuff that came out of this study, and there's a few points that I'd like to expand on in the future.
Well, tenuous.
Sorry, this is a tenuous connection with the dongle. So one of the things that we found was that we identified all of these potential, explanatory factors in our study, in our consultation with this community health advocate. But in this study, we weren't able to tease apart which of these factors really contributed to transmission. So in the future, I'm planning to do a more systematic study looking at how various metrics for economic and health disparities influence virus transmission. The plan is to do this in the context of SARS-CoV-2 and to look at how things like poverty indices, housing density, and health insurance access influence transmission. The idea being that if we can understand these effects, this can help us guide better policy. The second interesting thing that we kind of saw in this outbreak that I didn't have time to touch on much was that we noticed that these college-associated outbreaks are interspersed with these kind of community-associated outbreaks throughout the tree. And that suggests that there's at least some transmission from university outbreaks into these vulnerable communities. So I'd like to do some future work looking at the role that universities play in seeding new outbreaks of respiratory viruses, to see in more detail, for example, if we could update college vaccine guidelines and reduce risk for surrounding communities. Okay, so to recap, I started off today by telling you that viruses are constantly emerging and re-emerging. And I posed these two big questions to you. I told you that using H5N1 viruses as a model, we can capitalize on information in viral genomes to learn about how these viruses evolve within individuals and the factors that promote adaptation to new host species. We can also use these types of analyses to characterize how viruses jump between host groups so that we can design better surveillance and intervention practices. And finally, we can use a combination of genomic approaches with community engagement to learn something new about the factors that are driving an old virus transmission. And by using this information, we can make more informed decisions about how to update surveillance, outbreak response, and public health policy. And with that, I will take any questions.
Thank you, Louise. Very nice talk. So that's all. Are there questions in the chat? So I may have missed it, but how was transmission occurring between Arkansas and Washington?
Yeah, so I didn't touch on that. So this information actually came from talking to people in the public health department. Around the time that these outbreaks began, there were, this was kind of like right around the holiday season. And we know that there were some individuals in Washington from the Marshallese community who traveled to visit relatives in Arkansas. There's a pretty strong Marshallese presence in Arkansas. And so we think there was some travel history between
They brought it
locations.
back to Washington.
We think so, yes.
I had one other question before we open it up. So there's obviously an intense interest in trying to understand what's going to cause the next pandemic. Obviously, a lot of the work you're doing with avian flu is relevant to that. But are we ever really going to be able to predict that any? I mean, a lot of people thought that avian flu was going to be the next pandemic, right? And coronavirus sort of snuck in there. Is there any surveillance we can be doing, global, genomic surveillance, or that will help us predict what's going to happen, or is it just too random to predict?
I don't know that we'll ever be able to predict what the next pandemic will be. But what I think we can do is get a better understanding of what makes cross-species transmission more common, and we can put in place prevention mechanisms to try to prevent that from happening as much as possible. So I think what we can do is try to reduce risk and try to increase the knowledge we have so that if one of these viruses does emerge, we can catch it earlier. But I don't know that we'll ever be able to predict the next pandemic.
Okay, that's all. Other questions in the chat, or we can just open it up for the room here, and also people can unmute themselves and ask questions. This is a little bit of a new, this hybrid approach is a little bit awkward, but so any other questions?
I've got a few. Actually, why are vaccinated people susceptible to mumps? Is there something inherent about the population that the vaccine is maybe not as effective, or?
Yeah, there's a few hypotheses there. So definitely within the community, there's concern that they just don't respond to vaccine as well. That's not actually been tested. And so we don't really know
I was
if that's
wondering if
true.
you would test it, if you look at vaccine, levels or antibody levels in the vaccinated population.
You absolutely could. We have not. And nobody has really. I think that is worth doing. I think the other kind of hypothesis is that if you have like really intense contact rates, perhaps like your level of immunity you need to not get infected, with really prolonged contact is just higher than what you would need if you had kind of more diffuse contact. So those are, I think, the two main hypotheses there.
My other question is, I'm not much of a farmer, but I was wondering how you get cross species transmission between wild birds and chicken. I think the only chicken farms I've ever been to, they all seem to be like inside in these containers where they don't move around.
By design, right? I mean,
Yeah,
they're
by
segregated.
design. And I'm sort of wondering how they come into contact with wild birds to get those frequent transmissions.
Yes, that is definitely an interesting question. So we don't have much introduction of avian flu into US poultry populations for exactly that reason. So poultry are generally reared indoors in these large industrial complexes, which is sad for the birds, but like good for bird flu. In Asia, and particularly in Southeast Asia and China, it is really common to have much more like outdoor poultry rearing, particularly in areas where rice farming is really common. Wild or domestic ducks are actually often allowed to free graze in rice paddies because it's like cheap and economical. And so, what we think is that these migratory birds kind of like stop and then interact with those domestic birds. But it generally, I think, usually requires more like.
Do you know what the mode of transmission is between the birds?
So in birds, flu is fecal oral transmission. So a lot of transmission happens in these bodies of water. And if you surveil wild bird populations or just like common lakes where these birds stop over, you'll find like tons of avian flu.
So I have sort of a follow-up on that sort of line of questioning. So you had sort of hypothesized that you had long trains of transmission in the domestic bird population. Do you think it's more likely that you have a long train of transmission where you have the domestic birds that are actually retransmitting to the wild birds and then transmitting back to the domestic birds, because the domestic birds are not necessarily there for 10 years, right? But the wild birds may come back year after year after year.
Yeah, no, that's a super interesting question. So to answer the first part of your question, we do think that there is a little bit of transmission from domestic birds back into wild birds. I didn't show this data today, but we can estimate kind of the transmission rates between groups. And we see that the highest transmission rate is from wild birds into domestic birds. And then the second highest rate is domestic birds into humans. But right after that, there is some back transmission from domestic birds back into wild birds. And from talking to our collaborators, they've seen a couple clear examples of that. So I think that there is probably some back transmission, but I think in terms of the long-term circulation, you're right, these birds generally only live for about a year. But within that span of time, they can actually be moved around these really long geographic distances. So for example, a lot of these birds in Cambodia will be kind of raised on like a small farm. And then there's actually these people whose entire jobs it is to collect birds from different villages and take them to these like central locations where they all interact with each other. And then they get kind of spread out into these live bird markets. So there's just like ample opportunity for mixing of these viruses.
And that is really terrifying, by the way.
Yeah, it's pretty wild. When there's human outbreaks, you can go and sample kind of wastewater sample, or not wastewater, like wash water at these markets. And sometimes like 70% of samples come up positive for H5N1. It's pretty wild.
Any other questions?
I had a question. That was great, Louise. This is Seema. I had two questions actually. The first was sort of related to the H5 transmission and kind of what you guys were just talking about the longevity of how long you sort of see the diversity lasting. You sort of said it was short-lived, and I'm wondering, is that because are the wild bird markets, the poultry markets, the backyard poultry farms, are they vaccinating in Cambodia and Bangladesh or in Indonesia? I forgot the other site you were sampling at. Right, because that could then influence the diversity in which you're seeing in these populations and the reintroduction.
Yes, that is definitely something I'm planning to look at. So it's a little bit patchy. Cambodia does not vaccinate their poultry. Vietnam does, and Egypt does. there's some evidence that these so there's a whole lineage of H5 that is now like fully endemic in Egypt. And there's some evidence that there is antigenic evolution going on in these birds. So, it remains to be seen kind of how effective these poultry vaccines are, but definitely some countries are doing it more than others.
Yeah, no, that's really interesting. And then my other question was sort of to kind of follow up on your mumps data, which is really fascinating. But I love, and this is something I think we saw during the 2009 pandemic, was that you see multiple introductions, right? When you have a new virus emerging, it's not like it's seeded by a single infection. You really see multiple introductions that continually propagate of either from reinfection from the animal reservoir or perhaps going back and then coming back out. Those things are unclear, but I wonder how much of that is what you've mapped in mumps, was that these continual reintroductions, and can we use that as a way of trying to identify potentially what is the next pandemic by looking at when you start to see these sort of multiple reintroductions happening? Does that kind of make sense? I don't know if
Yeah, no, it does. I mean, I think if something kind of is continually like emerging and re-emerging, that, you would hypothesize that would be more likely to one day work and start transmitting really well. So yeah, I mean, I think as we get more and more sequence data, we can start to do this better and, identify kind of hotspots of emergence and hopefully then kind of tune in our surveillance mechanisms there. Okay.
John, did you want to ask your question?
I did, Lee, thanks. Hi, Louise, I'm John Williams, Pediatric Infectious Diseases. Great talk. I missed the first part because I had a meeting with my chair, but I heard the mumps part, which I love because as a pediatrician, I have seen mumps, and people are not aware that there's hundreds or thousands of cases a year because, as you pointed out, the M part of the MMR is just probably not as long lasting in its immunogenicity. So, I mean, I think your study was really fascinating because it's always a question in these big college outbreaks, is it sort of one person or multiple? And over this last year with COVID, I mean, Lee and I were both involved with COVID control here at Pitt. And what we were afraid of is just what you showed with the mumps, is that we were afraid that if we didn't control our college students, they would take it out into the community to the highly vulnerable people. So do you think it's feasible? Lee did work like this with COVID here to show, okay, the students had multiple strains. So it wasn't just one source, but it seems like, if you had the technologic pipeline, could you use this in like real-time detective work to say, okay, student health had 3 mumps cases. Oh, now it's 20. Quickly sequence and find out there's 2 sources, and then go to those sources and try and lock it down early. Do you think that's feasible?
Yeah, I mean, I think if you had a really good surveillance network and like a good sampling regime, you could totally do that pretty quickly. So the phylogenetic models that I showed you today are like very clunky and they're very accurate, but they're very slow. But you can actually do similar stuff with much faster kind of pipelines that you can run in a matter of hours. So yeah, I mean, I think if you had a good surveillance setup and a quick kind of turnaround on your sequencing time, you probably could do this pretty quickly. And I think during the pandemic, some people have tried to make those inferences more quickly, which is exciting.
Well, because
was it all based on the, sorry, John, I was just going to ask, is all the phylogenetics based on the SH or is it whole genome?
So we did whole genome, and we actually made a figure in the paper with what you would have learned if you had just done SH.
And what would you have got? Because that's the interesting question. Speed. SH gives you speed because it's only 57 amino acids, but whole genome, of course, is harder. So what was the outcome of that?
Yeah, maybe I should just show you.
No, honestly, we are going to speak tomorrow, and that's
okay.
That's in the weeds. Unless John's dying to know that answer, I'll chat with you tomorrow about that because that intrigues me.
Well, the short answer is that you would not have learned anything from SH.
Yeah, which is exactly correct. Yeah, that makes sense. Very interesting.
Yeah, so all of SH actually forms the entirety of North America, basically, is a large polytomy. So like literally everything's identical in SH. So you don't
Oh,
get
that's really interesting.
Yeah, it is super interesting. And again, that's where mumps is very useful because you can learn stuff because it's not the component of the vaccine is certainly not as efficient. You know, it just does not, people do not seroconvert as well with mumps. Mumps is different. If there's one component of MMR that will fail, it will be mumps, which is why the outbreaks are so interesting to follow. Anyway, sorry, John, I jumped over the top of you, but I was just dying to know, was it SH or what was the phylogeny based on?
Well, no, you highlight too the challenge, you know, I mean, at least as a negative strand virus, mumps exhibits very, very little recombination. But
Exactly.
for plus strand viruses like SARS-CoV-2, other coronaviruses, picornaviruses, you would have to do whole genome.
Yeah.
To look for the recombination.
Yeah.
For sure. Yeah, recombination introduces a whole new set of issues, but agreed.
All right, I think we're about out of time. So if there are any other questions, I suggest just emailing them to Dr. Moncla. And so thank you very much. Great presentation.
Great presentation. Thank you.
Yeah.
Thank you,
Great
everyone.
talk. Thank you very much. We'll see you tomorrow.
Thanks.
We did a lot more.